Good morning, good afternoon, or even good evening to all of you. My name is Florine Alès. I am the product manager for bioanalysis at Affinisep, and it's a pleasure for me to welcome you today to this new webinar on proteomics hosted by Affinisep. And today we will have the pleasure to welcome Dr. Kyle Bender from the University of Zurich as our invited speaker, and he will give us an insightful talk on quantitative proteomics of plant receptor kinase signaling. So Dr. Kyle Bender is a staff scientist at the ZIPFEL lab at the University of Zurich, where he studies mechanisms of receptor kinase-mediated signal transduction in plant response to the external environment. Throughout his career, Kyle has studied functional aspects of protein phosphorylation, including substrate specificity, regulation of kinase activity by autophosphorylation, and the allosteric activation of plant receptor kinases by phosphorylation. Currently, he is applying affinity and phosphoproteomics approaches to gain a quantitative understanding of how ligand perception by receptor kinases regulates receptor complex activation and downstream signaling. So before we have the pleasure to listen to Dr. Kyle Bender's presentation, I will just do a quick overview of Affinisep and the different products that we supply for sample preparation in proteomics. So for those of you who are not familiar with Affinisep yet, we are a French company created in 2013, and our team is mostly composed of scientists, experts in polymeric chemistry or analytical chemistry, with the objective to help analytical laboratories overcome limitations to their issues and find solutions to their problematics by providing technical expertise and also by supplying sample preparation kits. So as I said, we are experts in polymeric chemistry, and we manufacture all our polymeric resins from the monomers to the polymers on our unique technological platform, which is located in Rouen, in Normandy. And since we are manufacturers of all our products, we can control every aspect of the process to obtain specific resins that will have physical and chemical properties that are perfectly adapted to your needs. We can play, for instance, on the chemistry, on the particle size, or on the porosity. And if you don't find what you are looking for on a catalog, or if you need something very specific that you cannot find anywhere else, we can also work closely together to propose custom-made solutions. So we really innovate with you and for you. And in addition to our strong knowledge in designing resins, we also develop application for sample preparation and especially for solid phase extraction, SPE. So currently, our SPE resins are dedicated to three main application fields. The first one is food safety with a wide range of molecularly imprinted polymers, the MIPS, for the selective extraction of pesticides or toxins from various food matrices, so for instance, glyphosate or PAH. We also have a wide range of passive samplers for water analysis, so for the extraction of various contaminants such as PFAS, pesticides, drug residues, or endocrine disruptors from environmental water. And last but not least, we have also developed a product range for bioanalysis and proteomics with different ready-to-use kits that were specially developed for given application in proteomics, for instance, peptide desalting or peptide fractionation. So I will mostly focus on this last application field during my presentation. So for bioanalysis and proteomics, our latest innovation is a SPE membrane made of very small beads of sorbents, so much smaller than what is found in classical powder-based SPE devices on the market. And those tiny beads are tightly embedded together to obtain a soft, uniform, and mechanically stable membrane with high capacity for the extraction and purification of molecules of interest. And in addition to this high capacity of extraction, our SPE membranes also present various advantages. First of all, since the membranes are self-stand, we don't need to use filters or frits to immobilize the sorbent base, which greatly reduces the dead volume, which is already very low thanks to the fact that the tiny beads are very small and are tightly embedded together to leave very small volume between the different beads. So it's possible to elute your compounds of interest in very small elution volumes, thus obtaining highly concentrated eluates with high recovery yields and excellent repeatability. And if you also need to perform an evaporation step before the LCMS analysis of your sample, the time required for this step is also greatly reduced. And last but not least, our SPE membranes are available with a broad variety of sorbents, making them suitable for pretty much all application in bioanalysis and proteomics. These SPE membranes can be used to miniaturize SPE. So we currently propose 4 different formats for microelution and microextraction. We have, for instance, the SPE tips, the SPE in spin columns, the SPE well plates, and the SPE cartridges. And each of these different formats is available with different sizes. For instance, we have 10 microliter, 200 microliter, and 1 milliliter tips. We have 4 different sizes of spin columns, ranging from the micro spin columns to the 50 milliliter spin columns. 3 different types of SPE well plates, including 96 and 384 well plates for microelution, so for very small volumes of samples. And we have 3 different sizes of SPE cartridges. And in addition to this real flexibility of formats, our microelution tools also offer a real flexibility of capacity, since each format is available with different capacities. So for instance, with our SPE tips, you can use them to bind up to 100 micrograms of protein or peptides. You can use the spin columns to purify sample with protein or peptides up to 9 milligrams, up to 1.5 milligram for the SPE well plates, and up to 4.5 milligram of proteins for the SPE cartridges. So our formats for microelution offer both the real flexibility of formats and capacities, and can be used to purify sample volume ranging from few nanoliter and up to several milliliter, with protein or peptide amounts ranging from few nanograms to several milligrams. So if we focus now on the different formats and the different advantages that they can offer, starting with our SPE tips, it's important to note that one of the main advantages of our SPE tips is the fact that they are packed using fully automated methods. So we have an automate here in our lab that we call Jack, and that does absolutely all the work for you by packing the SPE tips using highly repeatable process, and thus avoiding the tip-to-tip variability that is often observed when you do your own stage tips. So you can stop wasting your time that you can use by doing more important things and more interesting things in your lab, and you can let Jack do all the work for you and prepare your SPE tips. Moreover, our SPE tips were specially conceived and developed for an optimal use by centrifugation, which is less tedious and painful than manual pipetting, so you no longer have to aspirate and dispense, and you can say goodbye to the tendinitis that is often observed for manual pipetting. And centrifugation is also more repeatable than manual pipetting, and it can help you save time by centrifuging several samples at the same time. And since we really want to facilitate the use of our products, we will supply ready-to-use kits that will contain not only the SPE tips, but also spin adapters and collection tubes for the simple centrifugation of the SPE tips, and then you collect your purified sample at the bottom of the collection tubes. So our SPE tips can be used by centrifugation, like we just saw, and we have a tutorial video available on our website that explains step-by-step how to use the SPE tips by centrifugation for peptide desalting, for instance. But our SPE tips can also be used by positive pressure. And in this case, you just have to add the solution from the top of the tip and then apply pressure to make the solution go through the tip. And thanks to this method of use, our SPE tips can be used in fully automated workflows with pretty much all sample prep automates that are available on the market. For instance, the Digest Pro from CEM, the Resolvex H200 from Tiken, or the EvoSep one from EvoSep. Now, if we focus on our other formats, they have, of course, their own advantages, starting with the spin columns that are used for high protein or peptide amounts. So they are very easy to use because you just have to centrifuge them in a microcentrifuge, for instance. And since they have a lid, they can be closed to perform direct incubation inside the spin columns, or you can use them to store your sample before the mass spec analysis, for instance. Our SPE well plates are used for high throughput experiments, of course, and they can be partially used. So you cover up the wells that you don't need, and you can reuse them later for another experiment without any consequences on the results. Plus, our SPE well plates are compatible with different methods of use, including centrifugation, positive pressure, or vacuum aspiration, meaning that they can be used with pretty much all devices that you already have in your lab. And finally, our SPE cartridges are used to concentrate large sample volumes in small elution volumes, and they can also be used with different methods, such as positive pressure or vacuum, and they are also compatible with pretty much all SPE device automates on the market. And since we use the same SPE membrane technology for all those different formats, including the SPE tips, the scale-up of the process or the change of the format is very easy without having to change or re-optimize the protocol. You can use the same protocol, but just changing the format depending on your needs and your throughput. So I've talked about our SPE membrane technology, and based on this SPE membrane technology, we have developed two product ranges for bioanalysis and proteomics. The first one is called Attract SPE Disc, which is a comprehensive product range composed of a wide variety of sorbents, either polymeric or silica-based. So you can find classical C18, HLB, or APS sorbent, for instance, in this range. So it's very chemistry-oriented. And the second range is called BioSPE, which is a new range of ready-to-use kits that were specially developed for given application in top-down and bottom-up proteomics, for instance, peptide desalting, peptide fractionation, glycopeptide enrichment, removal of magnetic beads after sp3 procedure, or purification of intact proteins. And today I will just quickly focus on our peptide fractionation kits, as this is the kit that Dr. Carl Bender used for his study. So for fractionation, we have developed the BioSPE PepFrac kit, which is used to perform reverse phase peptide fractionation at basic pH. So to reduce the sample complexity, for instance, for deep proteome sequencing or quantitative analysis. And this fractionation method at basic pH is orthogonal to the classical RPLC separation of peptides at acidic pH. So basically, after your digestion, you will separate your peptides in different fractions using an acetonitrile gradient, and you will obtain different fraction of increasing hydrophobicity, with the first fraction being the one with the most polar peptides, and the most hydrophobic peptides will be eluted in the last fraction. And this fractionation method is very useful to increase the number of protein that you can identify in your sample compared to unfractionated samples. For instance, you see on this chart that you have a 27% increase in the number of proteins identified compared to unfractionated samples. And this fractionation is very efficient with a good distribution of the peptides in all 8 fractions that you can perform with this kit, and a minimized fraction overlapping, with most peptides being eluted in only 1 or 2 fractions, meaning that the fractionation is very efficient. Moreover, this fractionation method at basic pH does not require an additional desalting step before the LC-MS analysis, contrary to fractionation performed by salts, for instance, on ion exchange resins. And our kit does not have any storage constraints, since it can be stored dry at room temperature, contrary to other fractionation kits available on the market, for instance. And finally, BioSPE PepFrac kit can be used to fractionate different types of peptides, including unlabeled peptides, but also TMT-labeled peptides and phosphopeptides. And this is something that Dr. Carl Bender will focus on during his presentation. So if you'd like to have more results or more information on our different kits, I invite you to have a look on our website, where you can find different posters on our different products. So peptide fractionation with the BioSPE PepFrac kits, but also peptide desalting with the Attract SP Disc Tip C18, so for automated or manual use, and also some application notes on the BioSPE Pure Pep Tips for peptide desalting. So if you have any question about this presentation of Affinisep, you can ask me now during this webinar by using the chat or question tabs, and we will answer them at the end of the webinar. But you can also contact me after the webinar if you want more information or have any question in the future by using my email address here. So now I will let Dr. Carl Bender present his work on quantitative proteomics of plant receptor kinase signaling. And if you have any question for Kyle during his presentation, you can also use the chat or questions tab, and it will answer you at the end of his presentation. So Kyle, the floor is now yours. You can now share your presentation and do your work. Thanks a lot.
Okay, thanks, Florine, for the introduction and for the invitation to give this webinar today. As Florine already mentioned, I'm a staff scientist in the Zipfel group at the University of Zurich, and our lab focuses on plant receptor kinase function in plant adaptation to the external environment. You see in the background there a picture of our institute. If you're ever in Zurich, I highly recommend you come and visit us. We're located in the Botanical Garden. It's a very beautiful place to be. Just a quick outline of what I'll talk about today. First, I want to give kind of a preamble to the whole talk and just try and understand the question, why is everything so green when you look outside? Next, I'll present very quickly what it is exactly that we do in the Zipfel lab here in Zurich. And then I'll give you a short primer on plant receptor kinase signaling that leads into some presentation of data on interaction proteomics of plant receptor kinases. And then finally, our deep phosphoproteomic analysis, where we've used the Affinisep BioSPE PepFrac spin columns to aid our work there. So when you look outside your window today, maybe depending on where you are in the world, you see a lot of green. The reason you see a lot of green is because our planet is heavily populated by plants. And this is no coincidence. The reason that we find plants almost everywhere we go is because plants are extraordinarily good at adapting to their external environment. The plants have this kind of unique innate ability to in situ respond and adapt their physiology to prevailing environmental conditions that allows them to survive and produce the next generation, even in some cases in extremely harsh environments. And so these kinds of things, this adaptability of plants is what really excites us in the Zipfel lab. And so the main biological question that all of our work is trying to address is, how do plants respond and adapt to a dynamic external environment? And we focus on a group of proteins called receptor kinases that you see, find my laser pointer, pardon me, that you see here. And our lab seeks to identify receptor kinases as well as the ligands that they perceive. We do that through bioinformatics approaches, through multi-omics approaches like the phosphoproteomics approach I'll describe today, as well as through genetics and physiology. We characterize the function of those receptor kinases, and our ultimate goal is to deploy those receptor kinases, and in some cases the peptides that they perceive, as tools to stabilize future agricultural outputs with food security in mind. So that's what we do. Those are motivations. And through this adaptive process that plants undergo, or during this adaptive process that plants undergo in response to their external environment, we find that both receptor kinases as well as externally derived and internally derived peptides are really key players in all of these adaptive responses. So I want to introduce you now to those proteins. So receptor kinases look a little bit like this. They have a conserved architecture where they have a large extracellular domain, a large or small extracellular domain consisting of diverse architectures. And those extracellular domains are tethered to a cytoplasmic protein kinase domain by a single pass transmembrane helix. These cytoplasmic protein kinase domains are monophyletic with the IRAC and Pele kinases that are well known in metazoan systems. The model organism Arabidopsis thaliana, the model plant Arabidopsis thaliana, encodes a family of roughly 600 receptor-like kinases, and among those, roughly 400 carry this receptor architecture, consisting of an extracellular domain tethered to a cytoplasmic kinase domain. Those extracellular domains come in a wide variety of architectures that we think allow the plants to perceive a number of different signals from the environment as well as from themselves. The other approximately 200 of those receptor-like kinases are so-called receptor-like cytoplasmic kinases that lack an extracellular domain but engage in receptor-mediated signaling processes in plants. The current model for how these signaling pathways are activated is roughly a simple on and off switch. So in the resting state, where the receptor is bare of its cognate ligand, we have our receptor kinase sitting in the plasma membrane associated with one or more receptor-like cytoplasmic kinases. And this is all in equilibrium with something that we call a co-receptor kinase that's recruited to the receptor kinase upon ligand perception. So when that ligand is perceived, we have a shift in the equilibrium that results in the formation of a stable receptor kinase complex in the plasma membrane. We don't know exactly the lifetime of these complexes, but we do know that around 20 or 30 minutes after signal perception, endocytosis occurs to clear the membrane of exhausted receptor complex. When those cytoplasmic domains come together, mediated by interaction between the extracellular domains of receptor kinases, they undergo phosphorylation. They trigger the phosphorylation of receptor-like cytoplasmic kinases that then function as executors of downstream signaling in those responses. Plant receptor kinases trigger a canonical suite of signaling mechanisms, some of which you might be familiar with. This includes the activation of MAP kinase cascades, the production of apoplastic reactive oxygen species, as well as the flux of calcium across both the plasma membrane as well as intracellular membranes. And these three, among a few other canonical signaling mechanisms, represent a core of activation mechanisms that are triggered by receptor kinases. Individual receptor kinases also trigger unique downstream signaling through other substrates, many of which remain to be identified, although the physiology is documented. In the ZIPFA lab, we use the plant immune system as a model to study receptor kinase function. The reason that we do this, the reason that the immune system in plants is a good model for this, is because we have a very clean on and off state, like I mentioned before. So in the absence of a microbial associated molecular pattern from either bacterium or fungus or other invading organism, we have release of these peptide elicitors, and these are completely absent when we grow our plants in sterile culture. So we can very rapidly and in a clean way trigger the on and off state of different receptor kinase signaling pathways. The receptors we study mostly contain leucine-rich repeats in their ectodomain. They use something called a somatic embryogenesis receptor kinase as a co-receptor. BAC1 that you see here in red is the founding member of this family in Arabidopsis. They perceive mostly peptides that are derived from conserved microbial proteins, but some of these also perceive peptides that are produced endogenously. You can think of these immune complexes in plants as kind of analogous to the myddosome in animals as a point of reference. With all that in mind, the kinds of questions that we're interested in, the kinds of questions that I'm interested in the lab are first, how is receptor kinase signaling organized within the cell? So what we're interested in asking here is understanding sort of what the membrane looks like inside the cell. Where are these receptor kinases located? Who are they located with in terms of other proteins, things like this. We're also interested in which ligands are being perceived and by which receptors, and in how that downstream signaling is orchestrated. And so those last two questions, the second two questions, are where our phosphoproteomics workflows are beginning to do some heavy work in the lab for us. But first, I want to touch on how receptor kinase signaling is organized in the cell, as this was kind of the birthplace of the quantitative phosphoproteomics in the ZipFill lab. So in order to understand how these kinases are organized, how they interact with one another, and how that might be involved in executing downstream signaling, we adopted a quantitative affinity proteomics approach of tagged receptor kinases, which are stably expressed in transgenic Arabidopsis plants. And our basic workflow is to affinity purify those GFP tagged receptor kinases. We use an SDS page gel as a kind of a cleanup step, and then we analyze those affinity enrichments in currently an Exploris 488 Orbitrap mass spectrometer. All of our downstream data processing and analysis is done in MS-Fragger, and then we use differential protein abundance analysis to identify either changes in receptor complex composition during a signaling event, or to look at steady state complex composition in the absence of any signaling event. And then finally, for those steady state experiments, we filter against proteome abundance under the hypothesis that proteins that are of high abundance are more likely to be contaminants in such affinity enrichment mass spectrometry experiments. So one of the things that motivated this work initially was to understand how this ligand perception event actually changes complex composition. And we had early hypotheses that phosphorylation of the receptor might be involved in some of these reorganization events. So we began by conducting experiments on some of our favorite immune receptors called EFR and FLS2. And we looked at those either in the absence on the left-hand side of 0 here, or in the presence on the right-hand side of 0 of their cognate ligands. And what we found, in fact, was contrary to our hypothesis. We actually see relatively little reorganization of the complex, or little change in the composition of the complex in response to ligand perception. So the proteins that you see annotated here in red are primarily members of this Circ co-receptor family that I mentioned previously. So while that wasn't a very exciting result for us, because this has been known for a long time that these Circ proteins are recruited to the receptors, it gave us a starting point to begin to look at how these kinds of complexes might be similar or different depending on the receptor kinase in question. And in these experiments that I show you here, we could already see some interesting trends with respect to the composition of these immune receptor complexes. So we expanded our collection of receptor kinases a little bit to look at a number of different receptors we were interested in the lab that have ectodomains of different types and were at least proposed to be involved in the perception of different kinds of ligands. And we performed this differential abundance analysis and filtering. Like I said, we did this for all of the receptor kinases listed. And for example, with our favorite receptor, FLS2, we find 99 candidate-associated proteins. Because we're a signaling lab, we're primarily interested in protein kinases. So after we did all this differential abundance analysis, we filtered for protein kinases. And what we found is that receptors with related function, so we're looking at FLS2, WAC1, and WAC2 here, these receptors that have related, potentially immune functions, seem to share a set of downstream signaling components. Yeah, that was quite interesting to us, and we're working on exploring this through genetic approaches now. One thing that was really striking, however, is that we identified a single receptor kinase, which I'm calling here COM-RK for common RK. We found this receptor kinase commonly in 5 out of 7 of the affinity enrichment experiments that we performed. So it's quite interesting to us, suggests that it might be a central regulator of these receptor kinase complexes. We tested that hypothesis with our favorite receptor, FLS2, that perceives the peptide ligand FLAG22. And we looked at some short-term and long-term outputs of that perception event. For short-term outputs, we looked at the production of reactive oxygen species in the apoplast, and long-term, we looked at changes in gene expression downstream of that perception event. And what we found is that in COM-RK loss of function mutants, we have a quantitative reduction in early signaling events as measured by a reduction of reactive oxygen species output in the COM-RK mutant, whereas later signaling events, here looking at gene expression, show a much stronger reduction in signaling output in response to that FLAG22 stimulus. And this overall suggests to us that this COM-RK receptor kinase might actually be part of feedback loops that are involved in amplifying the immune system during perception of these bacterial-derived elicitors. So I'll change gears now to move into our work on phosphoproteomics to talk about which ligands and by which receptors are perceived, and then how is downstream signaling orchestrated. And I'll just start with a bit of background on the current state of phosphoproteomics in the model organism Arabidopsis. So in 2020, a reference Arabidopsis proteome, including phosphoproteome, was published. In that phosphoproteome, they could identify roughly 40,000 class 1 phosphorylation sites, and they achieved that depth by fractionating at the organism level. So you see in the diagram here, the cartoon of the Arabidopsis plant, all of the different tissues that they collected samples from that they used for proteome analysis. There's 31 total tissues here, and in those tissues they find a combined roughly 40,000 class 1 phosphorylation sites. So this represents the greatest depth as far as phosphorylation goes for Arabidopsis, but it's important to keep in mind here that this is only in a steady state condition. There's no stimulation of the plants here. This is just documenting across developmental profiles which phosphoproteins or phosphosites are detected. More recently, in 2025, actually published by 2024, published by the Justin Wally lab in the US. They undertook an experiment to evaluate how FLAG22, this immunoelicitor I've talked about previously, alters the phosphoproteome in Arabidopsis. And they did this in whole seedlings. And in these experiments, they identified roughly 30,000 class 1 phosphorylation sites. They achieved this depth in their studies by using a tandem gradient on the LC that consisted of both reverse phase separation as well as strong cation exchange separation prior to injection into the instrument. So these are both really nice examples of good depth in the Arabidopsis phosphoproteome, but for us, it's not particularly practical because we're interested in understanding, first of all, rapid signaling events that are happening within minutes, let's say, of signal perception. And we're also interested in being able to compare different elicitors to one another. So you can imagine how fractionation at the tissue level or at the organism level might not be so practical with respect to cost and time. The second factor that we deal with is that we don't actually have a mass spectrometer of our own. All of our proteomic analysis is done in a core facility that's joint between the University of Zurich and ETH Zurich here in town. And we're not so closely located to that facility. So there's two issues that come to mind here for me. One is that we don't have access to customized LC setups, so we can't actually run tandem, reverse phase, and strong cation exchange. And second, because of our distant location to the Functional Genomics Center facility, we actually need simple methods that we can execute in our home lab so that we don't spend so much time traveling back and forth between the facilities where we need to grow our samples and where we need to do our analyses. And so with that in mind, we've been working over the last, I guess, almost a year now to optimize our phosphoproteomics workflows. And we've gone through 3 iterations of our workflow that I won't detail here, only to highlight that in the current state of our workflow, we've implemented these Affinisep BioSPE PepFrac spin columns, and we implemented those upstream of phosphopeptide enrichment by titanium iMac that's automated on a Kingfisher platform. And our samples then downstream of phosphopeptide enrichment are concatenated. And in all cases, we have loaded our samples onto EvoTips and analyzed those on an EvoSep 1 upstream of a timsTOF Pro mass spectrometer. Here's a snapshot of what these different experiments look like. I've categorized this by fractionation method, either where we did no fractionation, a biochemical fractionation that involved separating the samples into soluble or membrane proteins, or where we've implemented high pH reverse phase fractionation using the Affinisep kit. And what you can see here is that the Affinisep kit gives us a dramatic increase in the number of phosphorylation sites that we can identify in our samples. So this was really encouraging. We were quite excited about this when we when we got the results after our data analysis. When we look at how these different phosphopeptide preparation procedures compare, what you can see is that the majority of sites that we detect, either by the biochemical fractionation method or with no fractionation, are actually accounted for in our high pH reverse phase fractionation method as well. Although we do find a subset of sites that are missed by the high pH reverse phase fractionation approach. We don't know why that is yet, but that's something that we're currently exploring and are accepting in our analyses. When we look at analytical completeness, or how frequently we can identify a given phosphosite in any sample, what we find here is that we can identify roughly 2/3 of all of the phosphorylation sites in a plurality of the samples that we analyze. So we see 9 samples along the x-axis here. This is a subset of the samples that you see from the high pH fractionation set here. But in any case, we identify roughly 1/3 in the majority of samples that we measure. So this gives us good quantification on our phosphorylation sites. So how might we use this in a practical setting? So I just want to give a bit of background on the experiment that we conducted when we implemented this. As you probably know, plant cells are encased by a cell wall that's composed of different kinds of polymers, both cellulose and pectin, as well as other glycans. And during bacterial or fungal infection, many components of the cell wall are actually released into the apoplast, either through mechanical damage or through enzymatic activities derived from the invading organism. So it's no surprise, perhaps, then, that some of these molecules are actually perceived as damage-associated molecular patterns that can then activate immune responses to amplify plant immune signaling. In our lab, we're primarily interested in components of pectin. And for a long time, it was thought that a group of proteins called wall-associated kinases were the receptors for pectin fragments, or oligogalacturonides in this case. And a recent study from our lab using genetic analysis that undertook a CRISPR-Cas9 mediated approach to knock out all of these WAC-like receptors, the canonical WAC receptors in Arabidopsis, revealed that this doesn't alter responses to OG perception. So the plants are still fully capable of responding to OG even in the absence of these WAC receptor kinases. So this leads us to the question, what are the OG receptors then? And we used our phosphoproteomics pipeline to try and address that question. So here I give you an overview of what the data look like. We could identify roughly 32,000 sites in this experiment in total, and we had good quantification on approximately 27,000 of those sites. And from that set of sites that we could quantify, we identified roughly 500 differentially abundant phosphorylation sites in this analysis. Many of those sites were shared by 2 different elicitors, GAL-A3 and OGs. GAL-A3 is kind of the minimal epitope of the OG elicitor, so it makes sense that we see some overlap there. But we also see some distinct responses, suggesting that these elicitors might be perceived by slightly different mechanisms. So this gives us kind of a big overview of our approach here. But of course, as I mentioned, we're interested in understanding which receptors might actually be involved in these perception events. And so we filtered that data for all of the known receptor kinases in Arabidopsis, and we identified 2 proteins in our GAL-A3 treatments and 1 protein in our OG treatment, which actually matches one of the proteins from the GAL-A3 treatment, which makes sense. And we're now working on characterizing these as putative receptors for GAL-A3 and OG by other methods. But in addition to our ability now to use this approach to identify putative receptors for orphan ligands, we can also use this approach to characterize downstream signaling pathways, and this is something that I'm personally quite excited about. I mentioned previously that receptor kinase activation triggers MAP kinase cascades, and what we found in our GAL-A3 and OG experiments was that even though these elicitors trigger kind of a canonical immune response in plants, they seem to do so through potentially non-canonical signaling pathways. So in Arabidopsis, the canonical MAP kinases that are activated downstream of receptor kinases would be MAPK3, 6, MAPK4, and 11. And when we looked at our GALA3 data, we found actually a different set of MAP kinases that might be involved in the downstream signaling in response to GALA3 and potentially OGs as well. So this leads us to a number of interesting questions. First of all, how do different RKs activate distinct MAP kinase repertoires, assuming they do? And then how do those different activation events lead to similar downstream responses? With that, I already come to the end of my presentation. I just want to summarize what I told you today. First, I gave you an introduction and told you about how plant receptor kinases activate phosphorylation-mediated signaling in response to self and non-self derived signals. And our lab is interested in that in the context of understanding how plants adapt to changing environment. I showed you from our affinity enrichment mass spec approach that receptor kinases may organize into functionally related clusters, and that receptor kinases, which are common to these clusters, might actually mediate downstream signaling processes by mechanisms that we don't currently understand. And finally, I showed you how we implemented a simple high pH reverse phase peptide fractionation approach using the Affinisep BioSPE PepFrac spin columns to help us to identify potential receptors for orphan ligands, as well as to characterize downstream signaling pathways. With that, I want to just acknowledge some important people. Cyril is the leader of the group here in Zurich, and a satellite group in the UK as well. Yana Ordon is a postdoc in the lab who generated most of the samples that the phosphoproteomics experiments used. So she was instrumental in getting this workflow up and running in the lab. And she's now currently working on characterizing these putative OG receptors. Alvaro Fernandez, another postdoc in the lab, who's also involved in generating some of the samples that you saw today. Neftali Cruz is a new postdoc in the lab who helped with some hands-on work on the analysis. And Karan and Jack are postdocs as well, who have contributed to the affinity enrichment mass spec experiment that I showed you today, and some of the physiological and genetic analysis there. I want to thank particularly Antje Dittmann from the Functional Genomics Center in Zurich. She was instrumental in not only training me on the instruments there, but also in providing key advice to help us get our workflows up and running. Support staff in the lab here in Zurich, Lena Stransfeld, our lab manager, and Tamron Ellick, our horticultural manager. Former CZL members who contributed to the project, Laura Herold and Henning Mullenbeck, as well as IPMB support staff, and of course, all of the different funding agencies and institutes that have contributed funding to these projects over the years. And with that, I'd be glad to take any questions that you might have. Thanks for your time.
Thank you, Kyle, for this very interesting presentation. Thanks again for your time, Kyle, and for everyone who attended this webinar. And hopefully we will see you soon in a next webinar or in a next event. Thanks, everyone, and have a good day or good afternoon. Thank you all.
Thanks from my side as well.
Goodbye.